let us continue. We are finishing off our discussion of the spinal cord, spinal nerves and the spinal cord. When we finish this section today, we are moving into what the signals are that are sent by nerves in the spinal cord. Up until now, we've taken it for granted that signals get sent by nerves or through neurons. We don't really know what those signals are, so that's coming up. But let's round out our discussion on the spinal cord first. We left last class talking about reflexes. And reflexes you can think of as essentially the wiring or the circuitry for a feedback loop. Really? A reflex is really just a feedback loop. There's a sensor, something is integrated or processed, and then usually a motor neuron will execute some task. So reflexes and reflex arcs, the difference there is that the reflex arc is the circuitry that supports the reflex action. So the reflex is the thing that is accomplished. The reflex arc you can think of as the pathway that it takes, or the circuitry supporting the reflex action. And just like a feedback loop, you've got some sensors. So we used the example in class on Tuesday of you sitting on a thumbtack. Conveniently, the diagram here has a thumbtack in some kind of skin. There's a sensor that will detect changes in shape of some cells or whether the cells are pierced. There's pain receptors, stretch receptors. Something is sensing a change from normal. And that change from normal is relayed through the spinal cord where that signal is integrated. Remember, it can be processed directly by the spinal cord, which is shown here. Or it can relay that information to your conscious awareness if you need to think about it. You don't really need to think about stepping on a thumbtack or sitting on a thumbtack, though. And in processing that sensory input, you create a response. Here, a motor nerve is activating muscles that presumably will move you out of the way, or that you will use to reach and grab the thumbtack and remove it from where it's lodged. Those somatic reflex arcs are the things that you are aware of, that you have control over. So you're aware that you sit on something painful, and you're aware of the motion to move your hand and lift it. You're very welcome. Whereas autonomic reflexes are things that you're not aware of. So you're not going to be aware of changes in digestion or motility, changes maybe in heart rate, or of the glands that you're trying to activate. You don't control those. They happen, and you become aware of them, I suppose, as you start to sweat and drip all over the place. But you're not often aware of the autonomic arcs. This is an example of a somatic arc that you are consciously aware of. So this is a general schematic of a reflex arc. Let's look at a couple types, which you've done in lab this week, or you will do in lab this week. And even if you haven't been to lab, you've seen this example of a reflex, the stretch reflex. The tendon tap reflex that you've had done to you in the doctor's office when you were a kid. You sit on the edge of the bed, you swing your legs back and forth, and you use a little pointed hammer to hit your patellar tendon. That's an example of a stretch reflex. So what is sensed? How is this information processed? And what's the result? In the stretch reflex specifically, muscle spindles detect the quick, abrupt lengthening of the muscle. So imagine this. You're sitting on the side of the bed, and you can see the hammer here hit the patellar tendon. The result is that it essentially tries to pull the muscle apart. That's a pretty extreme description, but it does create this shock where the muscle is pulled taut at both ends. And the spindles will detect that. The spindles are a special structure in the muscle that monitors the rate of change of the length of the muscle. And if a muscle were to stretch too quickly, too far too quickly, that could damage the muscle. It can damage the joints, some other tissues, and we don't want that to happen. So fast stretch is something that we want to avoid, and the stretch reflex avoids it. The stretch reflex will avoid it by engaging the muscle to oppose the stretch. That makes sense. If a muscle's sitting at rest and then something sharply tugs at it, I don't want that to happen, so I can contract the muscle and prevent that tugging, to oppose that tugging or oppose that stretch. So the spindles in this case are fairly static. They're in the muscle and they're layered in between the muscle fibers. And they're always sending signals about the length of the muscle. That signal is interpreted at the spinal cord. And when an appropriate signal arises, a large enough stimulus arises, it will activate the muscles or the alpha motor nerve that innervates the muscle in question. So if there's a large stretch, I activate the nerve, that shortens the muscle. That's the stretch reflex. When you did this in lab, or you had it done to yourself, you probably noticed your lower leg jump a little bit, right? Your quadriceps didn't just contract, but your knee extended. There was movement. There's only movement not just because you're activating the target muscle, but because you are inactivating the opposing muscle. And this is an important concept that we don't often consider. It's sort of a supporting player in the stretch reflex. But there's only movement because the target muscle is activated and the opposing muscle is deactivated. Just think, if they were both activated, would your lower leg move? Probably not. If you contract your quadriceps and your hamstrings at the same time, nothing happens, ideally. So you need to deactivate the opposing muscles through a process called reciprocal inhibition. And this is usually the second prong of attacking any of these reflexes. So just beside the site where you see activation of the motor nerve that innervates the target muscle, there's a second neuron that's activated. And this specifically turns off the antagonist, or the opposing muscle. So the lower leg jumps because the quadriceps, the knee extensors in this case, are active, and the knee flexors are not active. Purposefully not active. So the stretch reflex is a classic example of a monosynaptic ipsilateral reflex. Monosynaptic being there is one synapse. The sensor activates a motor neuron. And ipsilateral meaning on the same side of the body. Clearly, when they named it, they weren't taking into account the reciprocal inhibition. There are two synapses, but we focus only on activation of the target muscle in this example. So stretch reflex, fairly straightforward and simple. Something is stretched, you contract it to stop the stretch. That was an example of an involuntary reflex. The tendon reflex, on the other hand, is voluntary. The tendon reflex can help explain how you can get deeper into a stretch, for instance. Or how you might fatigue quickly if you're lifting heavy weights at the gym. You might fade quickly. The tendon reflex is similar in principle. Something is sensed. In this case, there are sensors in the tendon of a muscle that measure how much tension is being applied, how much force is being generated by the muscle. And if that tendon is loaded, it will deactivate the muscle in question, activate the other muscle to allow movement. the opposing muscle to allow movement. So these are kind of like the spindles inside the muscle. The real name I'm not going to tell you because it's not up here, so don't worry about it, but it's a longer, odd, funny, complicated name. So the tendon reflex. If there is tension within a tendon, what happens? Tension within the muscle. In this example, I'm trying to flex my knee. By flexing my knee, I'm stretching the quadriceps. There's tension in the tendon attached to my quadriceps or my knee extensors. And so now that there's tension, what happens? Well, if I'm trying to flex my knee, I want it to flex. So I'm going to inhibit the extensors. I'm going to inhibit the muscle that's attached to this tendon so that it doesn't prevent me from moving my knee. What else am I going to do? I'm going to activate the muscles that are opposite from this tendon. And it might take a little bit to wrap your mind around the idea of the attached muscle, the opposite muscle, the agonist, the antagonist. But if you follow along and think about it, you'll get it in time. We want to activate the opposite muscle to allow movement. I'm trying to flex my knee. There's tension in the tendon of my knee extensors, the quadriceps. I want to make sure that I inhibit the quadriceps and activate the hamstrings. What this allows me to do is move my knee, flex my knee in the direction of my hamstrings, in the direction of my knee flexors. Again, we have an example of there's no movement in this case if the opposing muscle activates, so there must be reciprocal inhibition. Points 1 and 2 in the red square describe exactly that. I inhibit the opposing muscle and activate the muscle of interest, which allows me to swing my lower leg and flex my knee. So this isn't the best example to think about what I said at the start, that this reflex allows you to sink deeper into a stretch. Maybe imagine, not on the overhead, but imagine if you bent over to touch your toes. It's hard for some people, hard for a lot of people. It's difficult to do, but as you hang there, you tend to get closer to your toes. The hamstrings relax, the muscles in the lower back relax. And that's because of the stretch reflex that allows them to elongate, that inhibits them and allows them to stretch further and further over time. I'm trying to bend forward, I deactivate the muscles that would have me stand back up, and by doing that, they can stretch further and they can eventually reach and touch my toes. Both of these are fairly simple reflexes. There's one arc, one circuit. But there are a couple examples that are a bit more complicated, and this is the majority of reflexes in the body. This and the next one go together, the flexor withdrawal reflex and the crossed extensor reflex. These are a bit more complicated. They involve more nerves and more target muscles. But we can build on an example we've seen already. What if you stub your toe or step on a nail? I'm not just activating a muscle that will allow me to get out of the way. There's a couple things that need to happen. So the flexor withdrawal reflex is a response to remove a limb from danger. To flinch and remove a limb from danger. We want to avoid the injury, avoid excess pain. We want to get out of the situation that caused us pain in the first place. Let's look at what the flexor withdrawal reflex is. Now, I'll tell you up front, this is an example of an ipsilateral intersegmental reflex. So we know what ipsilateral means already. Same side of the body. Intersegmental is new, though. Intersegmental simply means that when the signal gets to the spinal cord, it doesn't relay out the same way it came in immediately. It activates higher and lower segments in the spinal cord. It spreads through the spinal cord so that we can activate other nerves and other muscles attached to those nerves. Intersegmental, activating multiple spinal segments. Well, the figure looks a bit complicated on the right. There are a lot of lines, and it might be even kind of small from where you're sitting, but the principle is very similar to the general schematic we presented at the start. We sense pain. That sensory information is transmitted to motor nerves that can remove us from that painful situation. Immediately, I'm going to activate the knee flexors at one level of the spinal cord to flex my knee and get it out of the way of the nail. But I'm also going to send that information up and down within the spinal cord to other areas that will activate the same muscle. What this allows me to do is make a more forceful and quicker withdrawal, because this is probably a pretty severe, painful experience. More segments of the spinal cord, activating more motor nerves, activating the muscle to a greater degree, generating more force, allowing me to quickly move my leg out of the way. And before we move on, just notice that it's one sensory signal that will activate motor nerves at that segment, motor nerves above that segment, and motor nerves below that segment. One sensory signal, multiple motor outputs. So the flexor reflex, the flexor withdrawal reflex, is a quick removal of the limb from danger. But this isn't all that happens, right? If I stepped on a tack right now as I took a step with my right foot, and all I did was flex my right knee to get out of the way, I'm not going to have a good day, right? I'm not going to stay upright. So I need to be able to also activate some accessory structures. And the crossed extensor reflex does just that. Well, now that you know what the situation is, the name crossed extensor reflex describes exactly what happens. Crossed, the other side of the body, extensors are activated in order to help support me in this situation. It maintains balance during the withdrawal reflex. This reflex is a contralateral intersegmental reflex. Contralateral, meaning opposite side of the body. Rather than leaving on the same side where the sensory signal arrived, we can activate muscles on the other nerves on the other side of the spinal cord to contract muscles on that side to help us hopefully keep our balance in this situation. So everything that I'm graying out or fading out a little bit is the withdrawal reflex that we just talked about. So let's move forward from that, knowing that that's active to get our foot out of the way. And now, how do we support ourselves to maintain balance in this situation? So that sensory signal doesn't only activate motor nerves on the same side of the body to withdraw the limb, but it activates motor nerves on the opposite side of the body, which will contract muscles of the knee extensors or quadriceps. That allows me to stabilize the leg. So I step on a tack with my right foot. Ouch, it hurts. I immediately want to withdraw this, but I have to activate stabilizing muscles in my left leg so that I don't fall over. I can flex my right knee, the one that's in pain. I extend my left leg to support myself and hopefully keep myself upright during this whole ordeal. So the crossed extensor reflex, contralateral intersegmental reflex. Both of those work together to keep you upright in a painful situation. So the words ipsilateral, contralateral, intersegmental, monosynaptic, those all make sense to you? So if I asked you what a contralateral intersegmental reflex was on an exam, you'd know. You would after reviewing. Okay, good. Let's come full circle and recap what we've talked about in this section. It's been a lot. Remember way back when we started this slide? From the top down, we left the brain. The first introduction to the spinal cord and peripheral nerves was the cranial nerves. The cranial nerves that arise from the ventral surface of the brain and some of the brain stem. There are 12 cranial nerves. You can remember what each of them are through this really helpful mnemonic. You also can remember whether they are sensory, motor, or both. Some say marry money, but my brother says big brains matter more. As we progressed inferior from the brain into the spinal cord, we looked at spinal roots. Oh, hold on. What did that happen? It just turned off. Okay, it's flashing. The projector's flashing, so it's turning on currently. Or it overheated. Oh, there we go. There's something. I thought, that can't be that confusing. It's a mnemonic device. It's a saying. We know the cranial nerves. It can't be a big deal. Yet, many questions. Okay, so we're good. We're back online. As we moved inferior in the body, into the spinal cord, we looked in general at the organization of white and gray matter, the rootlets and spinal roots, how they combine to form nerves. We acknowledge the one-way directional flow of information at the spinal cord proper. The dorsal spinal afferents receive sensory information. Ventral spinal efferents give off or send out motor information. If your name is Dave, it's easy. Or AMPS, anterior, motor, posterior, sensory. The roots combine to form nerves, and those nerves have generally both sensory and motor information. Last, what we just talked about today, that sensory and motor information achieves a goal. It does a job. It monitors the periphery, makes sure that things are working properly. If things are not working properly, it will hopefully do something about it, either quickly with an automatic spinal reflex, or perhaps you need to think about it a bit more using a cranial reflex that brings the sensory information to your awareness. We went through a few examples of what spinal reflexes are. You know ipsilateral versus contralateral. You know monosynaptic versus intersegmental. You know what reciprocal innervation means. Deactivating the opposing muscle. Awesome, good. And so that's the end of this section. And I'm waiting till the typing starts to fade because I'm going to switch slides. Looks like most of you have it. If not, find a neighbor, find a friend. Ask them what was on the slide. Because we are moving on. This whole time, we've talked about nerves, we've talked about the brain, we've talked about the spinal cord, we've talked about white matter and gray matter, and that things can be activated, things are sensed with really little understanding of what is prompting that sensation. What is being sensed, or how is it being sensed? What's the signal that's being registered? How is that signal created? How is it received? What we want to do in this section is understand those signals. How do nerves transmit signals? What are those signals? These notes are online, but I put them on this morning, so if there's some confusion, there shouldn't be confusion. They're all on Moodle. But you might not have been able to print them out right before. So we're going to look at a nerve, specifically a neuron, the base unit of the nervous system. Let's review the anatomy of a neuron to figure out how its structure supports function. Next, we are going to look at how the signal is generated. What is the normal resting situation? There is a membrane potential at rest. There is a charge in the neuron at rest. We don't have to know what membrane potential means yet, but we're going to learn that in a fair bit of detail. And then we are going to modify that membrane potential. From a resting situation, how does that charge change? Does it go up? Does it go down? Does it change quickly? Does it change slowly? Where does it go if it changes? This underpins every single nervous signal in the nervous system. Everything that you're sensing right now, the yawns that you're giving off, my voice droning on, it's all being registered through these nervous signals. So let's take a look at the base unit of the nervous system, a neuron. And there's a few structures of importance that we want to call attention to. Remember, neurons, like muscle cells in the body, are excitable. Those are the main excitable cells in the body. That is, they can generate a signal and respond to a signal. In a neuron specifically, I'm showing you one in the figure on the right. The dendrites receive upstream signals. They receive incoming signals. This is where the input arrives. We can call this the input zone. Based on how many signals arrive, the strength of those signals, and what they're trying to tell the neuron, you may or may not activate it. And the activation occurs at the axon hillock. This is the point of connection between the long projection of a neuron and the body and dendrites. The axon hillock is the trigger zone. The signal, if it's created, travels the length of the axon all the way to the end, the synaptic end bulbs, all the way to the bottom. And those will become important at the end of this section. So those are the main parts of a neuron that I want you to know. But there are supporting cells as well that play a role in the transmission of nervous signals. And those supporting cells are neuroglia. There are a wide variety of neuroglia. We're not going to touch on nearly a fraction of them. I just want to point out that a few are tasked with wrapping around the axons of some neurons. So the yellow bits are all neuron. The marshmallow-looking structures that wrap around the yellow bits are neuroglia. The marshmallow-looking bits are myelin in this case, and oligodendrocytes in the central nervous system create the myelin sheath, whereas in the peripheral nervous system, and what we're looking at here, are Schwann cells. They both create a wrapping, a coating around the axon. Myelin is a coating around the axon. We don't know why we want that yet, but we'll see why that's important coming up. Those two types of neuroglia create the sheath, the myelin sheath that extends the length of some axons. So for now, these are the important parts of a neuron that I want you to know. And it's the structure as laid out here that supports its function. Now, we're going to talk a fair bit about function moving forward. And as we go through and talk about it in class, you'll notice that there are a few other extra slides in your notes. And that's because we are going to watch a couple of really great 3D animation videos that summarize some of the slides that you have in your notes. So you have access to the videos. The links are included in your notes as well. You can watch them again if you want to, or they'll be on YouTube. And then you'll have supporting slides that kind of say or reiterate, back up the videos that we're going to watch. So we're going to watch these not sequentially in order, but we're going to take a look at least one of them today, and then the rest tomorrow and Tuesday. Now let's talk about a neuron at rest. Let's talk about the resting situation and how a signal is generated. At rest, most neurons are polarized. And this is an important characteristic of a neuron at rest. Polarized. What does it mean to be polarized? Polarized simply means that there is a difference in charge. Between two locations in a neuron, there is a difference in charge. It happens to be that there's a difference in charge between the inside and the outside of a neuron. It's not end to end, but it's inside to outside. And that difference in charge creates a potential for ions to move. You can imagine the simplest form of a polarized object that I think you might be aware of is a magnet, right? A magnet has two poles. It has a north pole and a south pole. It has a positive side and a negative side. They are of different charges. And a magnet is polarized. Its two ends have different charges. Well, something very similar happens in a neuron. Let's get rid of the magnet, and I'll put up a cell. This is a blocky neuron cell. The polarizing nature of a neuron doesn't occur end to end, it occurs inside to outside. So the inside is more negative relative to the outside for a variety of reasons that we'll explore. And that difference in charge allows us to do work, to accomplish a goal, to achieve some end. Now, a big question that you have right now is, why is it differently charged? And that's fair. That's a good question to have, and we're going to look at that in detail. But I do want to tell you right off the bat that the major players in creating this difference in charge are the cations sodium and potassium. There are other things at play, but these are major players that we're going to come back to a lot. And in a resting situation, there's a lot of potassium inside and a lot of sodium outside. There's very little sodium in the cell, and there's very little potassium outside of the cell. And so we create this distinction, or we create this division in the distribution of ions. And this is in part how or why the charge across the membrane is different. This isn't the entire answer. You're probably thinking, oh, these are all positively charged, so how does that make a negative charge? And that's fine. These aren't the whole answer, but I want to show you right up front the difference in distribution between sodium and potassium, and then you'll understand as we go through the discussion why that's important. So there's a resting situation that we want to maintain. High potassium inside, high sodium outside, and very little overlap or mismatch. Now, in that situation where you're just going to trust me at this point, the cell is polarized. From that starting point, I can either make the cell more polarized, the inside can become more negative than it is at rest. I can depolarize it. I can remove the difference in charge. The outside and inside have the same charge. Or I can make the inside more positive relative to the outside, which happens transiently in a special situation that we'll get to later. So hypopolarized in this case is lower than normal membrane potential. Depolarized means no membrane potential. Hyperpolarized means larger than normal membrane potential. When I say larger than normal, I mean more negative in this case. Hyperpolarized, more negative. The things that we're going to look at in the first video, and that we'll summarize after we watch it quickly, that make up the difference in charge are those ions that we talked about, sodium and potassium. There are other molecules. Phosphate has a negative charge, and that's limited to being on one side of the membrane, and also some proteins that carry negative charges. They're kept on one side of the membrane as well. And so the inequality of having some things on one side versus the other, the fact that there are sides, there's a barrier, and we keep things separate, is what allows us to create this differential across a membrane. So let's look at how this is created. And it just so happens that we're going to have enough time to watch one video, I think, maybe summarize it quickly before we call it for the day. This is a 6 to 7 minute video, but it does a fantastic job. If a picture is worth 1,000 words, then this 3D animation is going to be worth 10,000, 100,000 words. It does a really good job of trying to describe to you the normal resting situation.
We have the ability to perform intricate, complex movements, make quick decisions, consider creative, abstract thought, and communicate freely with others, because our neurons and muscle fibers have the unique ability to generate rapid electrical changes in your membrane. These changes are known as membrane potentials. An alteration in the electrical potential surrounding the cell membrane causes a neuron to produce a nerve impulse or triggers muscle fibers to contract. Neurons and muscle fibers are referred to as excitable cells. When the excitable cell is inactive, it is at its resting potential. Changes in the membrane potential may result in either a greater potential, impulses that either excite or inhibit further action, or an action potential, which results in some type of response. In a neuron, it becomes a nerve impulse, and in a muscle fiber, it stimulates contraction. To understand how a membrane potential occurs, we need to first consider the chemicals on either side of the cell membrane. The outside of each cell is bathed in a sodium chloride solution made up of positively charged sodium ions and negatively charged chloride ions. Inside the cell, in contact with the cell membrane, are positively charged potassium ions, negatively charged phosphate ions, and anionic proteins. Separated by the cell membrane, these chemicals accumulate charges on either side, creating a difference in electrical potential. The electrical balance on the membrane is altered when ions selectively pass through the cell membrane to the other side, generating a change in membrane potential. Ion channels, tube-shaped proteins across the membrane, allow specific ions to move through the membrane. The direction of ion flow through a channel depends on the difference in concentration of electrical charge action on the ion. Ions passively diffuse through a channel from higher to lower concentration. They are also attracted by opposite charges or repelled by like charges from other charged molecules. Thus, the direction of ion flow depends on the balance between both chemical and electrical forces acting on the ion at any given time. Typically, sodium ions tend to enter the cell through an open sodium channel, and potassium ions tend to exit the cell via potassium channels. There are four types of channels through which ions can pass. Leak channels are typically always open. The three types of gated channels, chemically, voltage, or mechanically gated, must open their gate to allow the passage of ions. A leak channel allows only a trickle of sodium or potassium ions to diffuse through the membrane. Leak channels contribute greatly to the generation and maintenance of the resting membrane potential. A chemically gated channel, also referred to as a ligand-gated channel, has a receptor associated with it. When its receptor is occupied by a chemical known as a neurotransmitter, the gate on the channel opens, allowing a specific type of ion to diffuse through. The gate will remain open as long as the receptor is occupied. A voltage-gated channel opens at a specific voltage, referred to as the threshold. For example, the threshold for a voltage-gated sodium channel is around -55 millivolts. When the membrane voltage increases from its resting state of about -70 millivolts to the threshold, the channel gate opens, and sodium diffuses through the channel to the other side of the membrane. Some voltage-gated channels open quickly, while others are slow to open, even though they may reach threshold at the same time. The speed at which they open causes significant changes in membrane potential. Some voltage-gated channels have an additional gate referred to as an inactivation gate, which temporarily plugs the channel so ions cannot pass through, even if their activation gate is open. Its purpose is to ensure that ions do not pass through the gate at an appropriate time. Mechanically gated channels open when pressure, touch, or stretch is applied to the membrane. They are found in sensory receptors, located primarily in the skin, to inform the nervous system of mechanical changes in the surface of the body resulting from conditions in its environment. An ion pump is a type of membrane transport protein. It requires energy to push ions from an area of lower concentration into an area of higher concentration. The ion pump most important in the maintenance of membrane potential is the sodium-potassium pump. Using ATP as its energy source, it is able to pump 3 sodium ions out of the cell and 2 potassium ions into the cell at the same time. Leak channels and ion pumps complement each other to maintain a constant electrical potential and level of ion concentration. The resting membrane potential voltage is determined by the concentration of electrically charged ions on either side of the membrane. It is established primarily by leak channels. There are more potassium leak channels than sodium leak channels, so more positively charged potassium ions pass out of the cell than positively charged sodium ions diffuse in. Also, sodium-potassium pumps move more sodium ions out of the cell than potassium ions in. Leak channels and ion pumps complement each other to maintain a constant electrical potential and level of ion concentration. As positively charged ions move out of the cell, they accumulate on the outside of the membrane, giving it a positive charge. The inside of the membrane is electrically negative because the negatively charged phosphate ions and anionic proteins lack channels, so they remain in the cell, and also because more positively charged ions move out rather than in. This establishes an electrical difference or potential on the membrane. Since the inactive cell membrane is negatively charged inside and positively charged outside, it is considered to be polarized. The voltage created by this distribution of ions is known as its resting membrane potential and is maintained everywhere on the cell membrane. The excitable cell remains in this state until it receives a stimulus.
I can see a lot of really interested faces. That's good. So you have other information in your slides on channels. That was something that was covered in this video really quickly. But just take away from this that the separation of ions, we specifically separate them, and they want to move back based on concentration from high to low, and then towards the opposite charge. we can harness that desire for those ions to move, to do a job, and send signals. We will look at how exactly that occurs in class tomorrow. Have a wonderful Thursday, and class dismissed.